Hey guys, welcome to my channel. Through this video, we'll discuss the complement system, activation pathway, biological functions, and complement deficiency. So, introduction. Paul Ehrlich coined the term complement and consists of serum and cell surface protein involved in defense against pathogens and tissues damage mediated by antibody. So, function may be the opsonization, chemotaxis, cell lysis, immune clearance, activation of inflammatory response. So, complement is a term group of protein found in serum in inactive form. Complement system, when activated, increase the strength of immune response. So, complement system is made up of 30 serum protein. The complement system, the protein system, and the regulatory protein. So, mesocyte is synthesized in the liver, while the minor sites is blood monocyte, tissue macrophages. Protein is in inactive form. When proteolysis, the larger fragments form the B part, while the smaller fragment known as the small a part. To participate in cascade reaction, exception 2A is a larger fragment. So in this, the larger fragment known as the small b, denoted by the small b, participate in the cascade reaction, while the smaller a fragment is always removed. And there is exception, that means 2 is larger fragment always. Complement system can be categorized into three pathway. That is, first one is a classical pathway, then alternate pathway, and the lectin pathway. So four main stages in activation of the any complement system is initiation of the pathway, formation of C3 convertase, formation of C5 convertase, and the formation of membrane attack complex, that is MAC. So classical pathway. The classical pathway is initiated by the antigen antibody complexes with the antibody isotopes IgG and IgM. So here is the whole pathway of the classical complement. Through the flowchart, we can easily understand what is this pathway. So here is a flowchart. When antigen antibody binds to C1, the C1 complex is formed, and then it splits into C2A and C4B fragment, and then it's combined to form C3 convertase, which is also called as C2,4B. And then with the help of a C3 hydrolysis, the C3B and C3A fragments is formed, and the C3B cleaves into the C5B and the C5A. Then C5B, C6, C7, C8 and C9 together form the cylindrical membrane attack complex, that is MAC, and swells and burst. So it is the classical pathway of the complement system. Here the C5 convertase is the C3B4B2 2A complex. The another pathway is the alternate pathway. It is a part of innate immunity, also called as the properdine pathway, and it is antibody independent. It is slower than the classical pathway. So here are the some details of the alternate pathway. With the help of the flowchart, we can easily understand. So here is a flowchart of all the pathway of the complement system. First one is lectin, then the classical and the alternative pathway. In lectin, the mass B1 and mass B2 combine to form through the help of the MBL, that is attached with the mannose. When binds with the antigen or microbes, it forms the C4 complex. And in classical pathway, the C1R and the C1S and C1Q bind with the antigen to form a C4 complex. Then C4 is split into C4A and C4B, and C4A is removed, and then the formation of the C4B2A, that is also known as the C3 convertase, and then it participates in the proceed reaction to form a C4B2A3B complex, and it's changed to C5, and then with the help of C5A is removed, C5B participate in the reaction, and ultimate it form the MAC complex of the cell wall of the any other cells. In alternative pathway, the C3 convert into C3B and C3A is removed, as we always say that the C3A is removed, which is a smaller segment, and C3B is a larger segment participating in the reaction. Then the factor B and factor D also supports to form a C3B B complex, which is also known as the C3 convertase, and then the reaction proceeds to form a MAC complex. The last one is the lectin binding pathway. It is independent of the antibody, as we can say that the lectin binding pathway and the alternative pathway is independent of the antibody. So first step is of binding of mannose binding lectin, that is MBL, to mannose residue on the surface of the microbes. To the MBL bound to a microbe, the MBL associated serine proteases, that is MAS P1 and MAS P2, will bind, and this complex will leave C4 and C2. The function of the complement system, that it facilitates opsonization, membrane attack, phagocytosis, inflammation, cell lysis, immune clearance, and the chemotaxis. Disease associated with the complement system. The complement system is an important part of the immune system and functions to prevent infection and disease. So, a common condition that complement system can be involved in include allergic asthma, anemia, cancer, kidney disease, macular degeneration, and the rheumatoid arthritis. So this is about the complement system, their pathway, that is alternate, classical, and the lectin pathway. So all about this. Thank you and keep watching.